For those of you who are new to Science Meets Music, I'm David Fitzpatrick, CEO, Scientific Director of the Max Planck Florida Institute for Neuroscience. And thank you for being here. Thanks to our generous donors whose contributions make all of this possible. And thanks again to the Benjamin School for sharing this wonderful venue for our program. All right, science meets music. When we say something meets something else, it implies that there's a space between them, a divide. And I've had a number of people tell me how surprised they are that we would put science and music together in one program. As if, these are two really different worlds that could never share the same stage. Well, for a number of you who I know are loyal fans of Science Meets Music over the years, I know that you appreciate how well science and music work together on the same stage, all right? And they do because they're both exceptional expressions of creativity, curiosity, and ingenuity that go far beyond the notes, far beyond the words, to capture our thoughts and imagination, to capture our sense of wonder. In short, both music and science stimulate our brains, inspire us, take us far from our daily routines, and give us a renewed appreciation for the talent, dedication, and determination that both musicians and scientists bring to their passion. Now, tonight, I am sure you will be inspired by the talent, dedication, and determination of three exceptional individuals who we are most fortunate to have as colleagues at the Max Planck Florida Institute. First of all, our scientific speaker is Dr. Hidehiko Inagaki, a research group leader whose neural dynamics and cognitive functions lab studies the neural basis for purposeful movement and how we learn to control the timing of movements, i.e., when to act. Obviously, understanding the neural control of actions is critical for all aspects of our behaviors and addressing a host of neurological disorders. And if you think about it in the context of music, learning to play an instrument brings timing and precision of movement into a whole new level of perfection. Tonight, Hidehiko will share with us his discoveries at Max Planck, Florida, about how networks of neurons in the brain work together almost like musicians in an orchestra to achieve this precision. But tonight is really extra special because we get to show by example another part of the magic link between science and music. This is the fact that many innovative scientists, including a number of Nobel laureates, have also been extremely talented musicians. In fact, Max Planck, our organization's namesake, known as the father of quantum theory, was an outstanding pianist. And his home in Berlin was a constant meeting spot for music-loving scientists with whom he would often perform. One of these was Albert Einstein, who was a superb violinist and loved playing pieces with Max Planck. In fact, Einstein has said he often played his violin while trying to work out a theory. And he even credited music with contributing to his greatest scientific achievement. He says, "The theory of relativity occurred to me by intuition, and music is the driving force behind this intuition." And the link between science and music continues to this day. And it's happening right here at the Max Planck Florida Institute, where we have many talented researchers who are also fantastic musicians. Tonight, you will get to hear two of the most talented musician-scientists perform together. Max Planck Florida Scientific Director Ryohei Yasuda on piano, and post-baccalaureate fellow Jordan Anderson playing the cello. Now, let me just tell you a little bit about them, and then I'll shut up and we'll get them out here and they'll play. So, Jordan is a scientist and a cellist from Washington State. He attended the University of Puget Sound, where he majored in neuroscience and minored in music. Throughout college, he performed solo, chamber, and orchestral works, took lessons from Alistair McRae, the principal cellist of the Princeton Symphony Orchestra. Jordan also taught private lessons and coached junior orchestra members at the Peterson Conservatory of Music. He combined his love of music with his passion for neuroscience by completing a bachelor's thesis on music cognition. Following graduation, he joined the Max Planck Florida Institute as a member of our post-baccalaureate program. This is where recent college graduates get hands-on experience with cutting-edge brain research that helps them make decisions about their future careers. I am delighted with how many of our postbacs decide that pursuing a PhD in neuroscience is the right thing for them. Now, our other musician tonight is Dr. Ryohei Yasuda, scientific director at the Max Planck Florida Institute, outstanding neuroscientist responsible for the development of numerous technological advances that have made it possible for the first time to visualize and manipulate molecular interactions within individual synapses, gaining fundamental insights into the complex molecular interactions that are the basis for a broad range of brain functions, including learning and memory. And Ryohei has received numerous awards for his research accomplishments. Sorry, Ryohei, I'm going to tell them a few, okay? Including the New Investigator Award from the Alzheimer's Association, the Research Award for Innovative Neuroscience from the Society for Neuroscience, and two of the most prestigious awards from the National Institute of Health. This is the NIH Pioneer Award and the NIH Transformative Researcher Award. Now, that in itself is absolutely exceptional. But add to that the fact that Ryohei is also an amazing musician. He's played piano for more than 40 years, and he also plays the harpsichord. He's often recruited to play the piano when he's speaking at scientific meetings. He's performed at Scripps La Jolla in San Diego, at the Federation of the European Neuroscience Societies, and many, many more. People want to hear him on the piano, as well as hearing about his science. And we are so fortunate to have him performing here at Science Meets Music. So please welcome Jordan Anderson and Ryohei Yasuda. Now for some science. It's my great pleasure to introduce Dr. Hidehiko Inagaki, who started his research group leader position at the Max Planck Florida Institute for Neuroscience in September 2019. So he leads the Neural Dynamics and Cognitive Functions Research Group. And as you will see, he's made, I would say, great progress in understanding the cellular and network mechanisms responsible for learning how to control the timing of movement. I'll embarrass him here for a minute or two. We had a huge meeting of scientists down the convention center in West Palm. We call it a symposium. And Hidehiko gave a talk there. And I had so many amazing scientists coming up to me and say, wow, you've really got a good one there. So Hidehiko received his undergraduate degree at the University of Tokyo, where he was already immersed in neuroscience, studying the anatomical and physiological properties of mechanosensory neurons in the fruit fly. That's right, in Drosophila. He came to the US and he pursued his PhD under the mentorship of Dr. David Anderson, who actually also gave a great talk at the symposium, where he studied the neural mechanisms underlying internal states, also in the fruit fly. He then moved on to the Genelia Research Campus of the Howard Hughes Medical Institute as a Helen Hay Whitney Foundation postdoctoral fellow, working with Dr. Karl Svoboda, studying neuronal mechanisms of short-term memory in the frontal cortex of the mouse. He is the recipient of numerous honors that I won't go through all of them, but he has received a number since he's been at Max Planck, Florida. The Peter and Patricia Gruber International Research Award in Neuroscience, the Cyril Scholar Award, the Klingenstein-Simons Innovator Award, Fellowship Award, and finally, recently, and this is fantastic, he was awarded the prestigious NIH Director's New Innovator Award, a highly selective award that recognizes early career researchers who demonstrate exceptional creativity and innovation in projects related to behavioral, social, or biomedical research. Exceptional creativity and innovation. What a great way to describe this tremendously talented young scientist. Join me in welcoming Hidehiko Inagaki.
Thank you, David. And we have such a beautiful music today. So now I'm going to talk about science, but something which may look like music, perhaps, in our brain. That's what I'm going to talk about today. Whenever more than two people interact or meet, something interesting could happen. They could become friends or come up with really new ideas, innovative ideas for business or many other things. And that's why interactions are critical for scientists as well. As David just mentioned, we had a symposium, which was a great conference organized by our institute, and we got a lot of ideas from there. So some of you may think scientists are nerds and never interact with other people, but that's not always true. Especially in our institute, I would say, we love to talk about new crazy ideas and also criticize each other to really shape the idea, to refine the idea. And this kind of interactions and synergy, because of the interaction, is the basis of the innovation and creative science. And this kind of synergy is everywhere for human activity. Like even for music, of course, there are a lot of musicians playing together different instruments. And when they are orchestrated together, there will be synergy or harmony, which will be really beautiful. And so synergy is everywhere for human interaction. But actually, it doesn't have to be human to interact for something interesting to happen. Even when animals interact, something interesting could happen. And even when two objects without any intelligence or without any brain interact, something could happen as well, which may sound odd. So I'm going to show you some example. So here, there are just two metronomes, okay? Just a simple metronomes. And this is a YouTube movie from a lab in Japan studying nonlinear dynamics, which doesn't matter now. But what they're going to do is they're going to start these two metronomes at different timing. So they are not synchronous at all. You can see that, and you can hear that as well. But over time, something happens. So please pay attention. I'm not going to say anything so that you can focus on it. Okay, I think everyone got it, right? It got synchronized. Maybe I should stop. Sorry, it's a little bit too long. Okay. Okay, I can't stop it. No, I thought it took longer for people to notice it. But anyway, I'm pretty sure everyone noticed that. Two metronomes got synchronized, which looks a little magical, right? But actually, this is not magic. Maybe some of you have noticed that these two metronomes are on a plate, and which is hanging from the ceiling. And so when one metronome moves, it could move the plate, which affects the other metronome. So actually, these two metronomes are physically interacting each other, and that's why they can get synchronized. If you do this on the hard floor, nothing happens. So it needs to be on the floating surface so that they can interact. So interaction can result in synchrony. And this lab is funny. They even try 100 metronomes to test whether they can synchronize. So it's totally random. At the beginning, I trim it because it takes so long. But you can see that, right? So interaction can create a lot of interesting things in the nature. And of course, animals, insects have brains, so they can do something more amazing. So this is a firefly in a part of Southeast Asia. So if you see just single firefly, it looks similar to a firefly in the US. But when there are a group of these fireflies, something really amazing happens. You can see a little different patterns of illumination here and there on the trees, right? Sometimes they just turn on, or there are some patterns from top of the tree to the bottom, and the patterns keep changing all the time. We can just keep watching this because it's so beautiful. And they have brains, of course, fireflies, but really small brains. I don't think they're smart enough to plan the illumination patterns for tonight, right? They're not talking about that. But the thing they're doing is watching each other. So they can see when the other firefly emits light, and based on that, they can decide when to emit the light. And this kind of interaction between fireflies become these beautiful patterns of illumination. So in nature, we can discover many different beautiful patterns, and usually that's because of the interaction between objects or animals or insects. So now let's talk about our brain. So in our brain, there are cells specialized for interactions, and these are called neurons. So if you zoom in to the brain, you can see cells with really interesting structure, and these are the neurons. So this is the cartoon showing the structure of the neurons. So there's a cell body, and you can see there are tree-like structure coming out. These are dendrites. So these are the places for neurons to receive input from other neurons. So these are like antenna. And there's a long structure coming out, which is axon, which is to send output to the other neuron. So basically, as I said, neurons receive input from hundreds of other neurons. And when there are enough inputs, there will be an electrical event or spike, something we call spike, which will travel all the way down the axon and send output to other neurons. So as you can see here, neurons are cells specialized for interaction. They receive input from many other neurons, process the information, and send it to the other. And there are electrical signals involved in this. In our brain, there are actually 86 billions of these neurons, 86 billions. It's a really huge number. The number of human beings on this earth now is around 8 billions. So we have 10 times more neurons in all of our brain. And like on this earth, everyone is talking to each other, trying to come up with something interesting, but something in 10 times bigger scale is going on in our brain. That's why we can do so many things. Okay? And if we can see our brain now, maybe it looks like this, right? So there are 86 billions of these neurons interacting each other. And if we can see electrical signal, we can see the blink of the neuron activity here and there. And now you're trying to understand what I'm talking, for example, right? And these neurons are processing that information. In this cartoon movie, all the neurons look kind of similar, right? But actually, that's not the case for actual brain. The neurons in our brain are really diverse, like how diverse human being is. And there is a structure of the brain which may be similar to orchestra. So in the orchestra, there are different types of instruments, right? Percussion, strings, and others, and which are arranged at different locations in the orchestra. And within the instrument types, there are different instruments. And similarly, in the brain, there's a structure. So if this is the mouse brain, which we are looking from the backside, of course, it's tiny because it fits the skull of the mouse. If we cut into the middle, we can see the internal structure of the brain. And different brain regions are shown in different color. So from the top, there's a neocortex, which is the region which is important for information processing in the brain. Under that, there's a hippocampus. You may have heard of this. This is a brain region important for many kinds of learning and memory. And there's a thalamus, which connects all these brain regions, and hypothalamus, which is important for feeding, social behavior, and many other innate behaviors. So as you can see here, there are different types of brain regions in the brain. And if we look into each brain region, like for example in the neocortex, we can see that there are many types of neurons within there. So neocortex has 6 layers. And in each layer, we have neurons with different shape, as you can see here. They look very different. So altogether, there are many brain regions in our brain, and in each brain region, we have diverse type of neurons. And in a mouse brain, it's estimated that there are something like 5,000 types of neurons. And most likely in human brain, there are even more. So I said there are 86 billions of neurons, but there are like huge diversity there as well. There's like maybe 10,000 different types of neurons talking and interacting with each other in your brain now. So it's a very complex system. Then how do we study this? So this is how neuroscientists have been studying the brain for many years, tens of years, which is to prepare a very thin electrical wire or glass pipette with electrical wire and insert it carefully into the brain. And when the tip gets close enough to a neuron, we can record the electrical signal from this neuron, the spike. So when this neuron tells something to other neurons, we can see the signal. But this only allows us to record activity of one neuron at a time. And if it's the human brain, then it's just 1 out of 86 billions. It's very small, tiny tip of iceberg. It's like just watching the one metronome in the crazy 100 metronomes YouTube movie, right? If you just see one, you can appreciate the synchrony. Or it's like just watching one firefly, right? If you just watch one, you can appreciate the patterns of illumination either. Or in case of orchestra, it's like just listening to one instrument at a time. Depending on the instrument, you may not be able to tell what kind of musical pieces they're playing. Of course, the technology to record activity of neuron in behaving animal is non-trivial. People made a lot of discovery, and many people won Nobel Prizes because of these discoveries. For example, by recording one neuron at a time in the brain region called visual cortex, people have figured out how the brain processes the information coming from our eyes. Or people have also discovered there is a map of this room or many other different locations in our brain as well, again by recording single cell at a time in animals. But as I said, neurons are specialized for interaction. And to study interaction, we need to record from multiple neurons at a time. So people have been trying hard to do that. And the simple solution could be just insert many wires. Then maybe we can record from 10 neurons at a time or so. But of course, we cannot scale this, right? Because if you insert more, you will damage the brain. So that's not a solution. So many smart engineers and scientists have discussed and tried many different ideas and came up with a very elegant solution, which is to use a technology to make the integrated circuit IC chip for computers. or more specifically, the CMOS technology used for computers and cameras. This technology allows us to fabricate a very small electrical circuit on surface of silicon wafer. So by using this technology, engineer has created a recording device, very tiny device with a lot of recording sites. So this is one of the examples of such device, which is called NeuroPixels probe, which was developed by a group of scientists and engineers more than 5 years ago. The most important structure here is this needle-like structure at the bottom, which is called shank. This is the part scientists insert into the brain. And this is very thin. It's 70 micrometer, which is thinner than our hair. And on this hair-thin surface, engineer has crafted a lot of structure called sites, thousands of them. And individual site corresponds to the wire scientists used to use to record single neuron. So basically, on this hair-thin surface, we have 1,000 different locations where we can record activity of neurons. So for the first time in the human history, now we have a tool to record the electrical activity of hundreds of neurons simultaneously, and this allows us to study the interaction between neurons. And it is something like this. So let's assume someone asks you to study the human dynamics in the Times Square. Of course, there are a lot of people, but you want to see how the conversation of the group of people develops over time. Then maybe the simplest solution is to just bring one microphone and record what individual persons say, which is similar to the old technology in the neuroscience, right? Recording one neuron at a time. But first, this is time-consuming, as you can imagine. And second, we cannot record many conversations at the same time, right? So NeuroPixel probes are like this. So there is a very long rope, 173 feet, which covers the entire Times Square. And every 4 inches, we have a microphone. So this kind of device will allow us to record all the conversation of people along this rope, like hundreds or thousands of people, and we can study the human dynamics there. So we have something similar to study the neural dynamics. And this is how the NeuroPixels probe look like. So compared to human finger, you can see how small it is. And because it's so thin, even if we insert it into the brain, it really doesn't damage the brain much. And also, it's very long with many recording locations. So we can record the neurons from top to the bottom of the mouse brain, all the 4 brain regions which I described. We can record all of them together, which was not possible before. People just used to study neocortex, thalamus independently in different animals. But now we can record all these brain regions and can study how these different brain regions and diverse cell types in these brain regions interact. So this is how the actual data we acquire in the lab look like. I'm basically showing the electrical activity from 30 different locations on the probe. If I'm going to show all the data, which are like 700 sites, it's going to be too busy. So that's why I just selected 30. And that's why you can see horizontal line, so which is the signal recorded at each location. And you can see vertical blip here and there. These are the spike. These are the signal coming out from individual neuron. So if the neuron tells something to other neuron, we can see this. And you can see that in this small segment of data, there are a lot of different spikes, right? So we can record the activity of many neurons. And this is how this signal develops over time in behaving animal. So you can see that there are spikes here and there at different timing with different synchrony. So this is a recording in a motor cortex in a mouse trained to lick after a cue. So we play PPP kind of sound, and they're trained to lick after that. And on the top, I indicated when we play the cue and when the animal lick. So you can see that before the cue, the brain is somewhat quiet, right? There are not so many spikes, there are not so many vertical lines. But just after the cue, it becomes really crazy. The density is so high and there are so many lines, and before and after the lick. So it's like in the brain, after the sound, like neurons are interacting with each other, saying there was a cue, okay, now we need to prepare for lick and execute the lick. There are a lot of interaction going on, communication, to process this lick following the cue. And if you see at the bottom, I'm showing the time scale, this happens just in 0.5 seconds, which is just half of 1 second. So in this very short duration of time, the neurons can interact and do computation to control the behavior. And we can visualize this process thanks to these new technologies. And doesn't that look similar to the musical score? This is Science Meets Music, so I really need to talk about music as well. In the musical score, basically what they describe is this, right? When each instrument should be played, right? And this data is showing when each neurons are active, right? So basically this is like we are acquiring the musical score of mouse behavior to lick after the cue. And if we do similar recording in the animal making decision, then we can get the data of the musical score of decision-making process. And similar to how people study the musical score to study music, we can look at this data and study how the neurons interact and process information. That's something we can do now. Also, we can represent the data in different way. So this is the internet activity on this Earth. Blue means low and red means high. And as the day and night cycle moves around, you can see that the peak of the internet activity move around. And we can see something similar in the brain as well. So this is a recording in our lab done by my student. And we are recording in the brain region called basal ganglia. It's a brain region which is important for motor control. Diseases which affect the basal ganglia, such as Parkinson's disease, Huntington's disease, affect our movement severely. So it's really important to understand how these brain regions process information and how they affect the activity in other brain regions. So here we have recorded from basal ganglia, which is showing blue shadow, and the thalamus, which is showing green. And this is how the activity looks like in a behavior. So when the neuron is not active, it's showing gray or blue small dot, but when the neuron is active, it becomes red. It's showing red. And this is recording in a mouse trained to lick after a cue. So you can see that before cue during the baseline period, like now, it's kind of quiet again, right? But just after the cue, brain becomes very active and there's synchrony of the neuronal activity just before the lick. So now, finally, we can really visualize this kind of process in the brain using new technology. And so this kind of recording of the neuronal activity during behavior is very important because this is a way for us to understand the information processing and the principle of the interaction between neurons. And in the long run, this could be translational for medical application because without understanding how the brain works, we cannot come up with a principle method to treat diseases. But actually, this kind of method to record the activity of neurons have a direct application, which is called brain-computer interface. So the idea is pretty simple, actually. We can insert the recording device, similar to what I have shown you today, into the brain, into the patient's brain, and record activity. And use that information to control computer, robots, or other devices. This is already helping many human patients with difficulty in movement to communicate with others or to move something. Okay, so today I have shown you that whenever more than two objects interact, something interesting could happen. There's a synergy. And actually, in our brain, there are cells specialized for interactions. And their interaction is the basis of our intelligence and all the cognitive function. And finally, we have a tool to record from hundreds and thousands of neurons simultaneously. So it's really now exciting, right? Because now we can start studying how they're interacting. Okay? And as I mentioned, interactions are critical for science. So I would like to acknowledge all the members in the institute and the lab, collaborators and fundings. Their interactions are the basis for our creative discoveries. And also, our institute has a lot of public events like this one. And in some of them, you can visit our institute and interact with scientists, which may lead to very better and big idea, perhaps. So please visit us. Thank you.
Well, did we bring science and music together tonight? Our thanks to Jordan, to Ryohei, and to Hidehiko for wonderful performances all around. And really, once again, appreciate all of your support. Thank you for continuing to join us for these. The next one is April 5th. And if you check it out on the website, you're going to find out that we're going to be doing a little more with science and music, bringing a director from Germany who actually studies the human brain and how it's involved with music. So please, hope to see you then. Take care.